welcome also to the very first joint seminars that we're holding in person again. I'm really excited about this. So it feels like the restart of the life after, which I hope will be even better than the life before, and certainly better than the life during. So this is really exciting. I'm told that I have to stand right here because we also have over 90 people that are actually joining this from afar or afar. Maybe they're just right up in this building, but at least joining online. We will hold the joint seminars in neuroscience as a mix for the next couple of weeks or months. So it depends on the speaker. We are giving the opportunity to the speaker to say whether they want to travel, whether they can travel or not. If the speaker is physically here, it will be both podcast and hold here. And if the speaker doesn't come, then it will only be podcast, because it doesn't make a lot of sense, at least to us, to sort of organize everything, check all of your records and so on, if there's nobody to be here anyhow. If you feel differently, please let me know. If you want always to have the opportunity to come here, congregate and talk to each other, please let me know, and then we can make that happen. Today is a very exciting moment with the BRI Magoun lecture for celebrating the existence of the Brain Research Institute, which goes back to Ted Magoun many years ago, who recognized that neuroscience actually was becoming a field that deserved its own being, its own entity, its own organism. Since then, we've become really accustomed to that, with many societies and departments and so on coming up. But the BRI, the Brain Research Institute, was one of the first sort of organized units to really look at that. So we're grateful to Dr. Magoun. But it's also a really exciting moment for the community to celebrate one of our superstars here at UCLA. And I'm very excited to be able to welcome Dr. Peyman Golshani as our superstar of the year. So he's presenting the 32nd annual Magoun lecture. And just as a slight parenthesis, we're again going to ask for nominations. And we're actually, that some of you who paid attention even to COVID think like, oh, we already had a 32nd Magoun lecture. So we actually doubled the lectureship. So we're now going to celebrate somebody in the beginning of the academic year and somebody at the end of the academic year. And Dr. Dean Bonamano was our first 32nd Magoun lecture. And now we have Dr. Peyman Golshani to start the new year with the 32nd Magoun lecture. I feel a little silly introducing Dr. Golshani, because I think almost everybody in this room and probably almost everybody online does not only know Dr. Golshani, but is actually actively collaborating with him. So looking through his CV is really impressive in the number of collaboration that he has. It's really sort of also celebrating the exceptional environment at UCLA, where collaborations are really fostered and highly regarded. So I was wondering what else I could tell you that you not only know that he's an amazing scientist, publishes really important and field-bending papers, has a lot of grant research support with almost all of you as co-investigators. So he started quite a while ago at Berkeley and got a master's not only in science, but also actually in English. He then moved on and decided to do an MD PhD. He moved to UC Irvine to do that. Got his PhD together with Dr. Ted Jones, who during that time moved to UC Davis. So then he spent a long time at UC Davis before he came back here before he came to UCLA to start here in his internships, and then moved through the ranks, through the academic ranks at UCLA. He's currently the inaugural Mazioda Chair in Neuroscience. And I just welcome him, congratulate you for the 32nd Magoun lecture. You get this amazing plaque that hopefully you can put someplace in your office. And so we need to stand here, and then Laurent is taking a picture. And that's just for the amazing archives. Congratulations. No, no. Okay. So without further ado, Dr. Golshani. Oh, and I didn't go through the slides that the staff so very nicely has prepared. Sorry, just one more second. So these are all the Magoun lectures up to today. You can also look at this on the website if you're really interested. And then this is a picture of Dr. Magoun, the award that I just gave to Peyman, and then a picture of Peyman. If you can't see him live here or online, there's always another picture available. So Dr. Golshani. We need to move over. And you can, if you have questions, if you're online, I will check the chat. So type your questions into the chat. And I don't know, do you want to have questions before or during?
I think I'll stop halfway through and then I'll ask for questions.
Okay, excellent. And Peyman has already warned me that since we're starting 12 minutes late with his lecture, we're going to go a little bit over. So just take that into account in your... There you go.
Thank you so much. It's really an honor and a privilege to get this award and to be here and speak to everybody. I'm greatly indebted to the collaborative atmosphere here at UCLA, specifically with the people that I started working with when I started my research here at UCLA with Felix, who mentored me tremendously, and then later Carlos Portera, and then later I worked with Josh and Alcino, and I've learned tremendously from everybody, and I would like to also acknowledge all the trainees that have come through and all the ones that are already faculty at different universities. I'm really I learned from my trainees all the time, and they train me. So there's a major question, I think, in neuroscience about how stable are the brain's representations of the outside world. We know the stability of representations from moment to moment in electrophysiological and neurophysiological recordings, but how stable are these representations over weeks, hours, days? And there's a lot of controversy about that. But more importantly, I'm interested in how stable are these representations of our internal cognitive processes. And there's also discussion about this, and many studies recently that show that the brain is highly dynamic. And it's almost, this is my one chance to quote a Greek philosopher, but Heraclitus said that no person ever steps in the same river twice, for it's not the same river and they're not the same person. And it's almost in that sense where everything is changing, everything is dynamic, nothing stays the same. And so, and I'm interested in these questions specifically. And so I'll start with a slide from Josh from 2002, where, you know, I think this was one of the first notions that there's a lot of, that things change and things are dynamic. He imaged spines in the cortex, in the somatosensory cortex with Carl Svoboda. And he again showed a great deal of the spines turnover, that nothing stays the same. Maybe some spines are there permanently, but spines turn over. And at the risk of showing a second slide by Josh, I think that the heavens will open up, I'll get struck by lightning. But if we record receptive fields, as you might expect, for example, in the visual cortex, receptive fields are largely stable. And if you record the same cells day after day, week after week, many of the cells really retain almost all their receptive field properties. So how does that sort of relate to the fact that all these connections are turning over? But that's not, it doesn't end there. There's work recently done in the olfactory, in the piriform cortex, in the olfactory cortex, where they actually use electrophysiology here, but recorded responses in the olfactory cortex and the piriform cortex, and the representations drifted. And eventually, after about 32 days here, it showed that essentially the representations were at chance, meaning that and these are just simple odor representations. So how does that work? I mean, we have very stable representations of our outside. When we smell, for example, an apple, an orange, whatever, the sensation is the same. So, but if these representations are drifting, then possibly downstream regions have to drift with it to keep our experiences the same. We know that our long-term memories can warp and drift and disappear with time, but what about our representations for more abstract entities like time, space? How stable are these representations? And what about the representations for the neural dynamics that link objects, concepts, and events in time? And these are just essentially our working memories that link events over just a few seconds. If you try to remember something over and over across days, even though this memory is very brief, how reproducible are these activity patterns? And is it possible that certain of disorders, neuropsychiatric or neurodevelopmental, maybe disorders of neural dynamic stability, either excessive stability or instability? And this is just a question that I'm positing and one that we'll try to answer. So I'll start out by talking about some work on the emergence and stability of working memory representations in hippocampus and secondary motor cortex, and then go on to talk about the stability of representations in disease, in models of autism and in epilepsy. And then if there's time at the end, I would like to highlight some of our work with new open source miniaturized microscopes that really led by our NSF Center and Daniel Aroni. So this work is by Yanis Taxidis, who has been a postdoc and project scientist in the lab and recently published in Neuron. We know about representations of space in the hippocampus. This is well known since for decades, and recently work by Howard Eichenbaum, the late Howard Eichenbaum, showed that in addition to space, the hippocampus can also encode time. The passage of time can be decoded from the activity of hippocampal ensembles. But again, how stable are these representations across hours, days? How do these representations emerge? And how does this actually come about? And Yanis was interested in this question, and so we decided to use two-photon imaging in head-fixed animals. These animals are head-fixed. Their cover slip is placed on the hippocampus. They express a genetically encoded calcium indicator, and the animals are trained while they are being imaged to perform a non-match to sample delay task. And here there's two different odors. If the odors match across the 5-second delay, the animal licks, and if the odors don't, if the odors don't match, the animals lick. If the odors match, the animals will lick. And the animals can be trained to do this task. And we essentially did this task because we were interested to see what happens to these representations over time as animals learn, and what happens as the animals perform a well-learned task, and how stable are these representations. With in vivo two-photon calcium imaging, you can actually do these recordings. You can follow the same cells across multiple days to weeks. And you can follow hundreds of cells. And this is two-photon calcium imaging of the CA1 layer. And we did this during learning and performance of the task. We can record not only from excitatory cells, but also from inhibitory cells. I'll talk a little bit about the inhibitory cells later, because we'll be using voltage imaging to record from those cells in particular. But here, from the excitatory cells, we can separate them by using an indicator for inhibitory cells and putting those cells aside. And then we can record the activity of the hippocampal cells and then deconvolve this activity. Every time there's a burst of action potentials, the calcium signal goes up, and we can deconvolve this activity and look And so what we essentially find is that we kind of separated this continuum of cells into two classes of cells. There are these cells which fire during specific odors, which we call odor cells. They either fire to odor A or to odor B. And then there's other cells which fire during the delay period. And here you can see these cells firing, let's say, around 2 seconds later, and these cells firing around 4 seconds later. And if you put all these cells and stack them and sort them, you can see that these cells make a sequence of cells that these fire to odor A, but not to odor B. And these sequence here, this is odor A, this is odor B. These cells fire to odor B in sequence throughout the delay period. This sequence bridges the two different odors. And if you take this data, it's fine to look at single cell information and then plot them. But if you really want to know what information is represented by the population, you have to use these decoders. Here, a Bayesian decoder was used, and we could decode the time that had passed. So there's information in the sequence to tell us how much time had passed and also what odor was represented. So there's all this information is present. And it's well above chance. Well, how does this representation emerge? If we actually look at this across training, what we found was as the animals go from essentially a little bit above chance to fully trained, performing around 80% behavior, we see that these odor cells are present before training and after training, but the delay cells accumulate so that there's more and more of them. And after the animals are fully trained, these delay cells now, there's a much larger percentage of them. And this correlates strongly with learning. So what happens if you just present the animal with odors over and over outside the context of a task? Well, you still have your odor cells and you have a few delay cells, but their number does not increase. There's no increase in the number of the delay cells. If we now use the benefits of two-photon imaging and look at the same cells over days, this is actually quite difficult to do within the hippocampus because it's a fairly dense mat of cells. But you can see there's these cells remain the same across days, and so you can line up these are sequence cells which were lined up across days. We find that these odor cells, a significant percentage of them were fairly stable. You could record them day after day after day, whereas the delay cells, if you look at this sequence that is shown here, and the cells are now sorted the same way, the delay cells show up, but the odor cells show up, but the delay cells become fuzzy, meaning that they tend to remap tremendously. So not only are the odor cells stable within a single session and the delay cells not so stable, they tend to fire not as reliably, but across days they're also not as stable. So here, this is an example of instability over time. And so whatever downstream target is receiving the hippocampal input needs to adjust to that. It doesn't go to chance. We can still decode time when the distance between days is, around 1 to 2, 1 to 3. It sort of tends towards chance, and by the time you get to day 5, the decoder is working at chance levels. And so to summarize this part, we can train animals to do this working memory task within the hippocampus. The animals have a representation of the odors and also the time that passes after specific odors. The odor representations are very reliable. The delay cell, time cell representations are not as reliable. And across days, there's a lot of representational drift within the delay cell population and not the odor cell population. And so one may posit that it could be that the GABAergic cells are regulating these sequences and actually are playing a role in the stability of odor versus time cells. We tried to record from GABAergic cells using calcium imaging. But these cells fire at very high rates, and it's difficult to follow them reliably, I believe, with calcium imaging. We are also interested in subthreshold dynamics, and wanted to know, for example, if the subthreshold changes that mediate the day-to-day changes are stable versus not. And you can't really do that with calcium imaging. You don't really get any information about subthreshold inputs. And so the way we had addressed this in the past, there was work done by Pierre-Olivier Pollack, who actually patched cells in vivo in the visual cortex. You can, I think we made this video with Josh together, I remember. And that's why we weren't able to show them in sequence, because he would show it and I would have to delete it. Yeah, or the other way around. But I think I'm actually guiding the pipette here. But he could guide his pipette to cells and record from cells in vivo, and then fill cells in the visual cortex and actually record from cells in the awake behaving animal. And so you can do this, which is actually fairly difficult to do. And you can never actually see the subthreshold dynamics across days, because you can't patch the same cell day after day. I think I tried doing that, it never worked. And what Pierre showed was that when the animals actually run, the cells in the visual cortex depolarize. This is due to the noradrenergic input. So we wanted to somehow get this kind of information with the hippocampal recordings and with the interneurons. So we decided to move to voltage imaging. And this has always been sort of out of reach for us and I think for all the neuroscience community. I remember talking to Lauren Luger at some point, and he said that this will never happen, that it's impossible to do physically. But Michael Lin at Stanford has designed these new ASAP indicators that are quite excellent, and we can record now from interneurons within the hippocampus using these voltage indicators. Blake Madruga actually built this wide field microscope, this epifluorescent microscope coupled to a very, very fast camera. We do this imaging at a kilohertz, and Yanis Taxidis did the imaging with Blake. And so now we can actually record from these interneurons. We don't get too many cells, a few cells, a handful of cells, like a max 3, 4, if we want to maintain our signal to noise. There's issues with bleaching, but I think we can overcome all these issues if you're really judicious about how much power you put on the brain. And so this is an example of what you get. You not only get the action potential firing of these interneurons and you see every spike, but you also get the subthreshold membrane potential oscillations. And this is, here we filtered the oscillations in the theta range, and we can actually clearly see that these cells fire along with the theta oscillations. Each of these colors is a single cell. The cells fire more when the animals are running versus still. And so what do we see when we actually look at these interneurons? We can do this across a number of trials. This is, I believe, a somatostatin positive neuron, and we did the same for parvalbumin positive neurons. We can actually see these, what I believe are hyperpolarization. So maybe this is inhibition of inhibition. These are all action potentials. This particular cell is firing, but we can also see the subthreshold modulations. And so what's fairly clear is that this is, there's much less selectivity for odor. We don't quite see these sequences that go into the delay period with the interneurons. It seems that these cells fire quite a lot. They're maintaining the equilibrium of the network, but they're not firing sequentially like the other cells. So this work is in progress. This is Yanis's sort of prize figure where he was able to follow the same cell across multiple days and record the membrane potential and action potential firing. So we're continuing this work and trying to understand how these receptive fields change across days in the interneurons as well as in the excitatory cells. And so this was in the hippocampus. So I was quite interested to see whether this instability that we saw in these sequences was limited to the hippocampus or could actually be found in the cortex. Arash Belafard, who is a postdoctoral fellow, he's actually a physicist. He joined the lab. He wanted to do theory. I told him he couldn't do theory. Yanis also wanted to do theory. I told him he couldn't do theory. And so they both became exceptionally skilled experimentalists. So Arash decided to use a different behavior. This is again, these are odors, except this is not a non-matched sample. This is actually a paired association test where the animals lick if you always have A and B odors as the first odor, C and D as the second odor, but if C follows A or D follows B, the animals lick. And if D follows A, C follows B, the animals withhold from licking. The animals learn this as well with 5 or even 10-second delays at fairly quickly within 4 to 5 sessions, which is, I always find quite remarkable. We really checked very hard to make sure the animals weren't cheating and doing an odor mixture task, but they are not. We measured a lot of different variables to make sure that was not the case. So we wanted to get a causality. So Arash used optogenetic inhibitors, the soma-targeted GTACR2, to inhibit the firing of these secondary motor cortex cells. He tried various other brain regions with mucimol and showed that the secondary motor cortex seemed to be important, but we verified it and got temporal precision. And so he inhibited these cells, and he could do so robustly, at least to a subset of these cells. He could inhibit them, either half inhibit or fully inhibit the cells. And what he found was quite interesting, is that when you inhibit the secondary motor cortex, the behavior of the animal really begins to suffer in the late delay period and within the second odor. They're almost untouched during the first odor, maybe slightly so. It's not significant during the first part of the delay period, but really in the late part of the delay period. So you may say, well, it's secondary motor cortex, it's motor preparation, but this is not a motor preparation task. The animal really does need to compare the identity of the second odor to the first. So this activity is sort of maintaining what that first odor is, and then the animal is making a decision based on that. So we were intrigued by this late delay period activity. Having shown that there's causality, Arash used mesoscopic 2-photon calcium imaging to really try to record from large populations of cells. This is a very large field of view that he was able to record. He used a strain of animal that expresses GCAMP in really all the excitatory cells. And he was also able to follow the same cells across days. It's actually easier to do in the cortex and the hippocampus. And what he saw was, this is a very small minority of cells will have very simple receptive fields. Like, for example, this cell fires to odor B and not so much to anything else. And this cell has a delay field. It fires to odor B during the whole delay period, but not so much to anything else. But these types of cells, which we were initially looking for, are extremely rare. What you end up recording are cells with very complex responses, what people I think call now mixed selectivity, or here you have cells that, let's say, fire to an odor B, but also to an odor D only after it comes after odor A, for example. Or they fire to a delay period to odor B, but also they fire to the first odor to a different stimulus. So you get these sort of combinations of things cells fire to, and this was by far the most frequent thing that we found. If you follow the activity of these cells, one theme that comes up is that as you train the animal, this is day one, the animal's untrained here, and then the animal acquires certain receptive field properties. This is one cell where the cell acquired delay-related firing that you could see come up during the later days and not so much during the earlier days. We quantified this by looking at the selectivity to the first odor. So during the first odor, early delay and late delay, and you can see that while the untrained animals really showed no selectivity, the trained animals, about 10% of cells had some selectivity to odor during the late delay period. So this does seem to be an emerging property. Decoders are very useful to look at to see what the whole population is doing. And Arash did this by using a long short-term memory RNN decoder. And this essentially, you throw in all the activity patterns of the cells, and it tries to, at best, predict what odor was presented at what time. So, for example, during the first odor, the decoder does extremely well. And then the decoder doesn't quite do as well during the late delay period, even though this is where optogenetically we had shown that the activity was important. Although, you know, we find about, you know, usually around 60 to 70% performance during this time, well above chance. In the novice animal, there's actually no decodability of the activity. So this emerges with training. If we now wanted to show the importance of M2 and compare it with M1 that actually Arash was able to record simultaneously, M1 really, there is no decodability of the odor during the odor period or after, but M2, you can certainly see decodability. So it does seem to be specific. And if you actually compare the correct trials and error trials, there does seem to be a difference, mostly during the late delay period. So again, arguing that this activity is important. So what about stability? That was sort of the whole point of looking at this activity. If you test and train a decoder on the same day, and you keep doing that across days after the animal is well trained, you can see that the decoder does pretty much fine. So within the same day, you can always decode the odor. You can always, during the late delay period, decode what happened, and it's fairly stable. You can also decode the choice of the animal. The choice, whether the animal licks or not, you can also decode that very well. And this tends to be stable. However, if you now start playing games where you train the decoder on one day and test on another, you get a sense for stability. Imagine, for example, that this activity was rock solid stable every time the same exact cells would get activated day after day after day. You could train the decoder on day 1, test it on day 7, and the decoder should do perfect, right? It should just about the same. This is not what happens. There's a great deal of instability. The decoder performance decays dramatically, and even during the odor period and the delay period, the decoder, again, over a few days goes to essentially a little bit above chance, but it takes a few days, meaning that there is some memory for what happened before, but it goes to chance. But most importantly, so you can train and test to various different days. And if you do that, what you find is that actually there seems to be this island of stability during the odor period, where if you train and test later, the performance is better. Not so much in the late delay, the part that we really cared about. There didn't seem to be much stability there. And so what emerges is that there's this mixed selectivity in M2, and again, the activity patterns that we record are not as stable as one would imagine across days. So I'll stop here in case there's questions, and to give people sort of. Do we have questions in the?
I think there were 2 comments more than questions.
All right, so I'll go on. Okay.
Here's my question. Yeah. So on your previous slide, when you're talking about previous one, yeah, there. So that, you know, when you train on one day, like try to code the other, you see that, you know, it's not going that well. Do you think that's due to population, which that like the cells that are being encoded in one day are different than the cells that are encoded information?
Yeah, that's what essentially is going on, right? Do I think there's population drift in the cells that are responding day to day, and that's essentially what's going on. If you take the population representation, then you can't really determine what was presented at all. So there's some drift in these populations. The animal's behavior is perfect, right? The animal's doing 90% behavior. These populations are drifting. It means that you don't necessarily need the same populations being active to do the behavior. All right, so yes.
In the first study, how do you distinguish between cells that are encoding for time versus memory of the first odor?
Yeah, so it's both, actually. This is a kind of we call it a multiplex representation. So you encoding the odor, but you're also encoding how much time has passed. So it's odor. It's not. If it wasn't odor specific, then it wouldn't be a memory. So it's a working memory representation. Yeah. Yep.
Are you able to sort of distinguish whether the drift is just time dependent, that in 24 hours it's being drifted a certain rate versus sleep dependent? I mean, did you ever look at morning versus?
We haven't done that. That's a very good point. Yeah.
Were the animals move their feet during that task?
So the first task they were on a ball, the second task they were in a tube, but in both situations the animals can actually, one of them they move a ball, the other one they sort of just move in this tube. So Arash has actually done recordings of the animals to make sure that locomotion isn't actually being the thing being represented. Same with Giannis, he took care of that. All right. Yes.
I was wondering, was there a reason why you looked at projections to CA1 over CA3 in the first study?
Yeah, so we to be honest, it's sort of everyone looks at CA1, and so we started there. It's a major output region, but we are looking at CA3 now. Stephanie Chung is actually, that's her thesis project, and she has essentially found the same types of sequences with possibly more delay cells actually in CA3. Yeah.
There's an online question from Daniel Tang, who is wondering what generalization metrics were reported for the LSTM decoder and whether the decrease in predictability is a result of an overfit decoder as opposed to an unstable representation.
Yeah, that's a very good question that Arash could probably answer better than I could, but yeah, I can address that later. Okay. Yeah. All right. So this notion of stability, these are all head fixed tasks that we've been using, and you could say, well, maybe you're not finding stability because the animal is not in a natural environment. These tasks are very artificial. And so if we want to look at disease, maybe we should try and look at disease in a more natural setting, working memory tasks that are more, the animal can sort of walk around. If you're looking at the hippocampus, the locomotion and the location of the animals is important for driving this type of activity. So it could be, I don't think it is, but it could be that if the animal was walking around and engaging in the real world, that these representations would be stronger. So that's when we made the move to actually try to look at freely behaving animals using miniaturized microscopes. At the time that we started, these were very expensive. And so we started with Alcino Silva and Baljit Kak, and then we brought Daniel on board as a postdoc. Daniel's now an assistant professor and really driving the creation, the testing of these new tools. He has an innovator grant from the NIH now and another brain initiative grant, so really doing extremely well, but we're all working together still. And at the time, Tristan Schumann and Denise Guy were postdocs in Alcino Silva's lab, and now they're also assistant professors at Mount Sinai. They worked with Daniel on a project to not only test these microscopes, but also look to see how the representations for place, for space change in a model of epilepsy. And so this was one of the first versions of these microscopes. These are, if you're not familiar with them, these are like big epifluorescent microscopes, except everything is smaller. Instead of an objective, you have a grin lens. Instead of a camera, you have a cell phone camera, essentially. And Daniel built the data acquisition devices and the cameras and put everything together. And with Denise and Tristan, they refined and optimized these tools. We open sourced these microscopes, and now I think over 2,500 microscopes are out there, and over 600 labs are using them. You can record in freely behaving animals really in any brain region you want, as long as you can tolerate the lens penetrating the brain and being over the cells that you're recording. So one thing that we really wanted to do was the wire tends to get in the way, so we created a wire-free scope. Daniel built a scope that is essentially battery powered and saves the data on a micro SD card on the head of the animal. We can do about 20 minutes of recording and get rid of the wires. The great thing that this does is it lets animals behave freely and run. This is not necessarily naturalistic for a mouse to run 25 feet back and forth across the length of the lab, but they can do this thing. And if you actually look at the spatial representations and image in CA1, the track is so long, you need 2 cameras to know where the mouse is. And so you can look at the activity patterns and look at the place-related firing in these ensembles. And we've sorted them so that you can see these place cells. But in these very large environments, actually place cells are not just simple fire in one location. Sometimes they have multiple fields. Sometimes they fire entirely along one direction and not another. So we can, with the use of the wire-free scope, we can get this kind of information. And then you can also put this data into decoders again, using decoders, and predict where the animal is. You can train the decoder either on the same day or on different days. And here the animal is running back and forth in black, but you see the decoder is hovering around it. Sometimes it makes a mistake, but this is an extremely long track, and this decoder was trained the day before. So there's some drift, but the degree of this drift could then be quantified in a disease model. So the disease model that we decided to use was one of temporal lobe epilepsy. I treat a lot of patients in the hospital with temporal lobe epilepsy. And I can tell you that they all have horrible memory. They have a lot of executive dysfunction. Not all, but I would say a great deal of them have significant memory impairment. People used to think it was the meds, but it's not only the meds, it's the fact that the hippocampus is involved. One hippocampus tends to shrink. So we decided to look at this in better detail. There was some work done previously that had shown place fields that were abnormal in a mouse model of the disorder. So we wanted to look to see how populations represent, whether population representations are altered and the stability of the representations in this temporal lobe epilepsy model. So we essentially took animals, injected pilocarpine, they had prolonged seizures, and we were able to track the same animal across. So the animal has a prolonged seizure, you treat the animal's prolonged seizure ends, but this animal will then go on to develop, just like a temporal lobe epilepsy patient, spontaneous seizures once a day, twice a day, sometimes more. And then you can look at the representation of space in the hippocampus of these animals. And what we found was that this representation actually becomes rather imprecise. In the controls, you can see very precise representations here on these are fairly clean place cells that fire only in one direction and not the other when the animal runs back and forth. But in the epileptic, they're broader, and they're actually, if you look from trial to trial, they're less stable. So even within a single session, they were less stable, and we quantified this. Stability was lower, and the precision of the firing was lower. But if we now again look to see how stable these representations are, the stability actually is dramatically, this is in the control at 6 hours. You can essentially see, if you see the same thing here and here, that means the representation is stable. If it becomes smeared out, it's less so. In the controls, you can clearly see the same place cells firing in the same locations. In the epileptic animal, this is already smeared out at 6 hours, and by the time you look at in 7 days, you can still see a trace of where the place cells were. Some have remapped, but in the epileptic, this is completely remapped. So this is an example of a disease situation where there is actually these cognitive representations have become less stable. And we can quantify this using correlation coefficients, and then also with decoders, you can see that this decoder does fairly well in the control animal when it was trained on the previous day. It can follow the animals in white, the decoders in red and green, depending on which direction it thinks the animal is going. So it does a pretty good job. The epileptic one is just like totally lost. It's just jumping around all over the place. Sometimes it's green, sometimes it's red. It's mixed up. And so this is an example of what we can do looking at the stability of these representations. But is this true also in autism? Ping Zhao is actually looking at a model of autism, the CatNap2 model, where there's autism and epilepsy together. And one thing we wanted to focus on was not a representation of space per se, because this is autism, but a representation of social interactions. And so she recorded animals from the nucleus accumbens because we think that the nucleus accumbens plays a big role in social reward. And we think that's like, you guys are all here because social interaction is rewarding for us. And it is also for the animals. They prefer interacting than interacting with an object or with Zoom. So she imaged these animals in the nucleus accumbens for 7 minutes, and then was able to also image them with an object and quantified interactions. And here, one animal is wearing a miniscope and the other animal is not, and she's imaging them in the accumbens, and she's recording the activity with the miniscopes. And she quantified the percentage of cells whose activity correlated with the social interactions using a receiver operating characteristic study. And then she did shuffling controls to make sure that that was indeed statistically significant. And so here you can see an example of a cell which was activated by social interactions. These overlap, and a cell which here is negatively correlated. You can't see it that well, but in this cell is neutral. And so about 20% of cells were activated by social interactions in the accumbens. This was far greater than what we found in the prefrontal cortex or in CA1. So it seemed to be that this accumbens may be a hub for social interactions. If we use decoders and try to decode social interactions, when the animals actually, so when this O high will turn on when the decoder thinks the animals are socially interacting. And so it does fairly well. It's not perfect, obviously, but it does fairly well. And we can quantify how well this decoder does against the social target and an object target. And it's about 70% correct with the social target in the accumbens, but it's at chance with an object. If we do this in the prefrontal cortex, we get similar results. But the truth be told, in the prefrontal cortex, we're imaging many, many more cells. And if you do the controls, which I'm not going to show, the accumbens for the number of cells that you use in the decoder does better. And the hippocampus does terribly. It's at chance. It can decode what the object interaction, but we think that's because the object is in one location and the hippocampus has information about location. And so what about stability of these representations? So I've showed you working memory representations that are unstable and place cell representations that become unstable with disease. What about the stability of these ensembles? Either if they're completely stable, then everything recurs. A social cell will be a social cell the next day, et cetera, or there could be turnover. And so Ping did these experiments and compared the activation of these cells across days and overlapped them. So she's sure she's imaging from the same cells. And she gets around, it's much more than chance, around 20% of cells are socially activated in the accumbens, and the same with the prefrontal cortex. If it's you have a 20% chance if you're a social cell to be a social cell the next day, and the next day, and the next day, so forth. And so if you look at this across multiple days, there's a very little chance that a cell is a social cell across all 6 days. So there seems to be considerable turnover there as well. And so I brought this up because we were interested in autism and wanted to see whether these representations are impaired in autism. We use these catnap animals, and they have hyperactivity, they have epileptic seizures, and they have deficits in social interactions. We looked at their activity, and again, there is a decrease in the social interactions in the knockout animals, and the average duration of interactions is less, but the number of socially activated and inhibited cells in the accumbens was around the same. When we look at the activity of these cells during social interactions, you can see that there are socially activated cells and socially inhibited cells in the catnap animals. But if we use a decoder, the catnap animals actually do worse. And so they're better than chance, but they do worse than the wild types. So we're actually looking at the stability of their representations as well to see if they're impaired. So I will, since my voice is getting tired. Your brains must be really tired. I will just end by giving a plug for the new open source tools we've developed. This is by we, I mean Daniel has developed, and our center is promoting. There's, I think, 600 of these new generation miniscopes. Many of you have used it. It has a much bigger field of view, where he has built, and we have collaborated with Michel Basso to test very large field of view heavy microscopes in monkeys. We have built an electrophysiology capable microscope to do concurrent electrophysiology and imaging together. Everything digitized through a single coaxial wire. We were using this. This is work by Sung-Won Her and Karen Safarian in the lab, who are working together to do electrophysiology in the cerebellum and also do imaging in the ACC at the same time during social interactions. We find socially modulated cells here. And I'll end by just showing some work by Blake Madruga in the lab, who's building a 2-photon miniaturized microscope, which has much better resolution than our 1-photon microscopes. We can image dendrites and axons, hopefully, with it. And this was our first prototype. It requires lasers and hollow core fibers and such. This is the actual design of the new scope. There's a scanning mirror here, which scans. These are custom objectives that he designed with Julie Bentley and Daniel Aaroni and Matt Strawman. So this actually is the largest field of view 2-photon miniaturized microscope. This is how it looks like. And we imaged corn because it gives a very pretty picture, and it's a standard imaging thing, but it's too bad we're not interested in corn. And we are also able to image neurons in vivo. This is a recent image that he got in collaboration with Mega Segal and Alcino Silva, imaging one of their mice in retrosplenial cortex. So the microscope works. I'll stop here. A plug for miniscope.org, where you can get all the miniscope related information. And thanks to the lab. This is an old picture before the pandemic, just to make us feel bad. And again, thanks to all the collaborators across the years, graduate students, postgraduate. I've mentioned people. There's many undergrads that took part in these experiments over the years, and I appreciate all their help and also the funding sources as well. Thank you.